Hi, my name is Raeesa Gupte. Um, Let me just start with a little bit of background about myself. I am originally from India, where I did my bachelor's in pharmaceutical sciences. And then I moved to the United States, did my master's in cell and molecular biology, did my PhD in pharmacology, and for about the past close to 3 years, um, I was a postdoc at the University of Kansas Medical Center. So for my PhD, I worked with ion channels in the brain and how they affect stroke. Hello, I see some more people joining in. Welcome. Thank you for being here today. I was just starting to introduce myself. I was just saying how my PhD research was focused on brain stroke and mechanisms in which we can modulate the ion channels in the brain to maybe prevent neuronal damage after a stroke. And then for my postdoctoral research, I sort of shifted gears a little bit and moved into the field of traumatic brain injury. So, stroke and traumatic brain injury still both come under the umbrella of brain injuries in general. And they have very similar mechanisms that go wrong in the brain when someone gets either a stroke or a traumatic brain injury. But there's obviously differences in how these injuries are acquired, and then how the outcomes differ and things like that. So kind of similar, but still a little different. But in the overall realm of brain injuries in general. I've always just been interested in the brain, so that was just something that I didn't want to stray too far away from for either my PhD or my postdoc research. Um, so traumatic brain injury is hi, hello people, welcome. We are just getting started with the introduction. So traumatic brain injury is the name is pretty self-explanatory. It's a physical force or a trauma to the brain which results in an injury. So you can think of a physical trauma as anything that results in an impact to the head. So things like motor vehicle accidents or falls or assault or person to person violence, stuff that can result in a strike or a blow to the head can cause traumatic brain injuries. Hello, hi. Oh, hi. Thank you for being here. Um, so yeah, so that's one aspect of it. The other aspect of it is that sometimes it doesn't necessarily have to be a blow to your head that causes a traumatic brain injury, and that is where concussions come in. So concussions have been in the news quite a lot lately, given the whole sports injuries and do they lead to concussions and how does that affect um, athletes in the long run and stuff like that. So concussions are essentially the mild form of traumatic brain injury. So you can either get a concussion by like I said, due to a blow to the head or by hitting your head against something, or it can just be because of the movement of your brain within the skull. So as we all know, the skull is this bony protection that our head has, so to protect the skull, I'm sorry, to protect the brain. And the brain is essentially just like floating around in cerebrospinal fluid inside the skull. So if there's any sudden jolting movements or sudden acceleration, deceleration type of movements, your brain can slosh around or jostle in your skull, resulting in the brain getting bruised because it either strikes the inside of your skull, which is a hard bony surface, or because laws of inertia or laws of motion dictate the way and the biomechanics in which the brain moves. So different kinds of tissues in the brain are probably going to be moving at different velocities. So when there's like this sudden jostling kind of a movement, there's tearing and shearing of neurons, and that leads to neuronal damage and concussion related um, symptoms. Hello, I see some more people joining us. Hi, welcome. I was just getting started with hello. I was just getting started with like a brief introduction of what TBI or traumatic brain injury is and what the difference is between concussions and traumatic brain injuries. There isn't really a difference, it's just concussions are a type of traumatic brain injury or a milder version. Hello, or a milder version of traumatic brain injury. And while a more moderate or severe kind of injuries might be due to a physical impact or blow to the head, a concussion might not necessarily be because of a blow to the head. It might just be because of the biomechanics of the brain and the way that the brain is sitting inside the skull and sudden jostling movements causing the brain to slosh around within the skull and get bruised and cause tearing and shearing of axons, leading to neuronal damage. Hello, people. Um, uh, so that was just like a brief definition of what traumatic brain injury is. As I was just mentioning, some of the causes of a traumatic brain injury could be motor vehicle accidents, or falls, or person-to-person violence or assault, um, or even sports injuries these days. With football and soccer and hockey, all of these kind of contact sports resulting in impacts to the head or sudden acceleration deceleration sort of movements. Um, hello, that that cause damage to the brain or bruising to the brain. One other type of injury that is actually gaining some ground now is blast TBI, or a traumatic brain injury because of explosions. So usually soldiers or veterans on the field have suffered these kind of injuries. In fact, I think in the US, almost 10 to 20% of veterans who had been deployed to Iraq or Afghanistan seem to suffer from blast induced TBI sort of injuries. So that's another kind of injury that is starting to become evident and starting to be being studied right now. So what how is it that traumatic brain injury or TBIs are detected in the clinic? How is their severity determined? How is the damage that they have caused determined in the clinic? So for the severity, very commonly there is a scale which is called the Glasgow Coma Scale that is employed to test how severe the brain injury is, which is essentially a questionnaire looking at a few different functionalities. So one of them is eye movement, the other one is verbal ability, and the third one is motor function. So based on these three kind of broad overall categories, physicians assign a score to the patients that come in, and this score can vary from 3 to 15, with 3 being a very very severe injury where the patient is unconscious, almost in a coma, and 15 being very very mild, where there might be some little daze or confusion that the patient is experiencing, but otherwise they're fine. They are conscious, they can talk, they can follow instructions and stuff like that. So a lower Glasgow Coma Scale score represents a more severe traumatic brain injury, and a higher Glasgow Coma Scale score represents a milder traumatic brain injury. So concussions, like I was saying, are mild TBIs. So usually a concussion would have a score of 13 to 15 on the Glasgow Coma Scale. So that was how physicians determine the severity of a traumatic brain injury. Hi, hello people. Thank you for joining and being here this Saturday. Oh, there's hearts and stuff. Okay, thanks. The other part that I wanted to discuss was, how is the damage that has occurred in the brain detected in the clinic? And for that, there is two main techniques that are employed. The first one, oh, thank you. Glory Microbe says you expressed it very well. Oh, thanks a lot. That's the goal. Um, so how is the damage that the brain has incurred measured in the clinic? So there's two diagnostic imaging tests for this. One is CT scan, or computed tomography, which is a series of X-rays that forms a detailed picture of the brain, and you can then view, tissue damage or bleeds and skull fractures and all of those sort of things in a CT scan. And that is usually going to be the primary neuroimaging method that is used in the clinic by physicians when someone comes in with a TBI. Are you a research student? I am not currently a student. So I finished my PhD in 2015, and then I was doing a postdoc for about 3 years. My postdoc has ended recently in September of this year. So I was still doing research up until very recently, and now I'm kind of busy writing up all of the data that I have got to publish into studies and publish it in scientific journals. So not really a research student, but I feel like we're all lifelong students once you get into science, because you just like to learn about new stuff all the time. Um, so yeah, one of the primary methods of detecting brain damage after a TBI, as I was saying, is a CT scan, which gives very detailed images of the brain using X-rays. And the other method that is also commonly used is MRIs, or magnetic resonance imaging. And what this does is it uses a very powerful magnet and radio waves to take pictures of the brain. So you might essentially see very similar things in a CT scan and an MRI, but an MRI is going to be more sensitive and show you more detailed stuff with better resolution. So this is usually used a little later on, not as the primary diagnostic step, but afterwards, if they don't see any improvements and are trying to figure out what other things might still be wrong, or what they may have missed the first time. One of the caveats to using MRI is, like I said, it uses a very strong magnet. So if someone has like metal rods in their body, you cannot use an MRI on them. So when patients first come in and doctors don't know whether they have metal implants or pacemakers on them in their body, they that's why they try and avoid MRI as the first step, and that's why CT scan is the diagnostic imaging that's preferred in the clinic. Which leads to treatment. So once physicians have determined how severe the injury is, using the Glasgow Coma Scale, and there's some questions about how effective the Glasgow Coma Scale is and whether it misses things and stuff, but that it is kind of the most prevalent or widely used questionnaire to detect or to determine a patient's initial level of functioning after a traumatic brain injury. Hello everyone, I see a couple more people joining in. Thank you for being here. I was just talking about how a traumatic brain injury is diagnosed in the clinic, and moving on to how once doctors have determined the extent of the damage, how they might how they might proceed with the treatment. So right now, unfortunately, there is no FDA approved treatment specifically for traumatic brain injury. So a lot of stuff that is done in the clinic is mostly just trying to take care of all the symptoms that take place after a traumatic brain injury. So if there's bleeding, then doctors are going to try and stop that. If there's clots, they're going to try and remove those clots. If the brain is fractured, I'm sorry, if the skull is fractured, they're going to try and figure out, how to join join it back together. Um, so yeah, it's just a lot of symptom management of TBI in the clinic. There isn't really a cure or a treatment for it. One other thing that, in terms of drugs that clinicians use is stuff to reduce the swelling on the brain, which is often due to accumulation of fluids or water in the brain. So to remove all of that, they might give patients diuretics, which is something that increases the production of urine in the body. So it'll help drain the fluids out of your body for management of brain edema. One other thing that happens after a TBI is there is an increase in uh, pressure inside the brain after a TBI. So there's increased intracranial pressure, as they call it, and this needs to be relieved because otherwise it starts compressing all of the important structures in the brain, such as the brainstem and the cerebellum, which control all of these important involuntary functions of our body, such as breathing and heart rate. So to avoid compression and damage to these very delicate precious structures, it's very important to relieve this intracranial pressure that builds up after traumatic brain injury. And one way of doing this is simply to kind of drill a hole in the skull to relieve that pressure, to have some place where that pressure can escape to, as opposed to just filling up inside the skull. So I see a question here. What can't the CT scan see which MRIs can? What can the implant patients do in such a case? This is interesting. I'm not sure. Okay. So there's other techniques, other neuroimaging methods that maybe patients with implants or pacemakers can make use of. So stuff like PET scans, which is positron emission tomography. Sorry, is another way in which stuff can be. You can have detailed pictures of the brain. So if someone can't get an MRI, then you can get a PET scan or a CT scan. Um, the MRIs are also, as I said, slightly more expensive because the equipment is huge and it takes quite a lot of maintenance on those equipment. So CT scans are the more preferred form, and they can almost detect everything from skull fractures to swelling to bleeding in the brain pretty well. Um, so since there is no real treatment for traumatic brain injury, that's where the research scientists come in. So we want to figure out a way to reduce all of the damage to the neurons. Oh, you're welcome. Reduce all of the damage to neurons that happens after a traumatic brain injury. So let me back up a little bit and start with some of the cellular mechanisms that go wrong after someone gets a traumatic brain injury. So in the brain, we have these very controlled gradients, electrochemical gradients that are there, which maintains sodium ions on the outside of a neuronal membrane and potassium ions inside, leading to positive charge outside and a slight negative charge inside. And all of maintaining this balance is critical to proper firing of action potentials of neurons and just adequate release of neurotransmitters and all of that stuff that needs to remain balanced in the brain for it to function properly. When someone gets a traumatic brain injury, the pumps in the neuronal membrane that are responsible for maintaining this proper balance of ions and electrochemical gradients start misfunctioning, and that leads to a lot of sodium ions just coming inside, rushing inside the cell, firing action potentials, leading to, and when sodium comes in, a lot of calcium also gets to come in. And when there's too much calcium in the cell, the calcium is like a double-edged sword. It, um, it's a good thing because it's a kind of like a secondary messenger within the cell. But if there's too much of it, it overwhelms the cell. So usually the mitochondria in your cell, which are known as the powerhouse, they are responsible for generating all of the energy that of a cell. The mitochondria are pretty good at handling increases in calcium, but when someone gets a traumatic brain injury, there's just too much calcium and it overwhelms the mitochondria, leading to all sorts of things that can go wrong. You get lots of inflammation, there's oxidative stress, all of this imbalance in the electrochemical gradients leads to a massive release of the excitatory neurotransmitters of the brain, which is the main one is glutamate. So when there's too much glutamate, there's a lot of it leads to more neuronal death. So there's all these mechanisms that go wrong in the cell, and it's important to understand them because only when we understand these mechanisms can we design proper treatments to treat the ensuing neuronal damage after a TBI. So that's what is the focus for a lot of research labs around the world, trying to understand the mechanisms that go wrong after a TBI and what ways are there for us to fix them, to develop better therapies. So for the research that I was doing, a lot of times to understand these mechanisms that go wrong, it's very hard to do this in humans first, and then there's also a lot of ethical considerations. So you can't just start enrolling human subjects and start testing them because you're going to get into a lot of trouble if you do that. So to pinpoint the exact mechanism that goes wrong and to get around all of these ethical issues, animal models have been very, very important in understanding mechanisms of traumatic brain injury. And there's a lot of different models that you can use and there's a lot of factors that also need to be considered for this based on how much it's going to cost, what exactly is the research question that you're asking, the biomechanics of the brain and stuff like that. So it's very popular to use rodents. Can you just clear about the blood-brain barrier? Yes. So there is the blood-brain barrier is this. It's essentially what it says. It's a blood-brain barrier. It's this barrier of cells that keeps all of the toxic substances that are in the blood from reaching the brain. And it does so with the help of specialized capillaries of the blood cells, as well as glial cells in the brain called astrocytes and microglia. And these the blood vessels of the brain are specialized because they have these endothelial cells that make up the lines of the blood vessels that are very close together. They are very tightly knit, and they have proteins called tight junctions that help keep them together. And all of these mechanisms that are in place help protect the brain from unwanted stuff that may be swirling around in your blood, and allows only nutrients, glucose, and some some substances that are only mainly fat soluble to cross into the brain. So it's very important that you have an intact blood-brain barrier. But after a traumatic brain injury, there is extensive damage to the blood-brain barrier, as we can well imagine, which can also again. So another thing that the blood-brain barrier does is it keeps immune cells from the blood and the periphery out of the brain. So when there's damage to the blood-brain barrier, all of these immune cells rush to the site of inflammation, and themselves also release all of these chemokines and cytokines and other inflammatory molecules, adding to the inflammation that is already happening in the brain, making things worse a lot of times. So would TBI patients get better or worse with time? So TBI outcomes, whether patients improve or get worse, usually you improve, um, but the rate at which you can improve depends on a lot of things. It depends on age of the patients. It depends on the gender of the patients. It depends on um, some of their genetic factors, their medical history. There's a lot of things that are in play over here. So, for example, if someone is older, they're not going to heal as fast, and older patients with TBI have a higher rates of more. There's higher rates of hospitalizations and deaths in older TBI patients as compared to younger ones. There's a lot of research that's going in recently now about how gender affects TBI outcomes, and that's something that I've been writing up a review on recently. So I'm super interested in this kind of thing. We are not exactly clear how gender affects whether the rate at which someone might get better after a TBI. There's some human studies that show that women may do worse. That some that show that women may do better than men. A lot of the animal literature has shown that women might do better. So that led to the speculation that there might be some hormones, female hormones, that might be protective in nature. And there was this big clinical trial that was conducted on progesterone, which is one of the main female sex hormones, but that did not show very clear benefits and had to be stopped. So it was kind of a failure. So yeah, we still are not very clear about whether men do better or whether women do better after a traumatic brain injury. It can also depend a lot of times on the kind of questions that you're asking. So, for example, if someone is looking at depression after traumatic brain injury, a lot of times more women report depression after a traumatic brain injury as opposed to men. But if you're looking at something else like motor function, there might be mixed results on that, and no clear indication more of motor function and mortality and stuff like that. Um, yes, yeah, yes. There is some regeneration that also occurs. So yes, can get better over time. It takes a lot of neurorehabilitation, physiotherapy, speech therapy, all of that sort of stuff that needs to happen for people to get better after a TBI. You wanted to ask about depression next. Oh, okay. Thank you. Thanks for your best wishes. Um, okay, so where were we? Oh, yes. So animal models of TBI, yes. Um, so rodents are usually the most preferred animal model in any kind of research, not just neuroscience research, given that they're very easy to maintain, they reproduce quickly, they're cheap, um, and because they also share a lot of similarities with humans. So there's one thing that I am always surprised about is that mice share almost 98% of their DNA with humans, which seems like it would get something really crazy to me. Almost every protein that humans have, mice have a similar one or a homolog of that protein. So you can see why it would be relevant to study human conditions in animal models using rodents such as rats or mice. People also use pigs for TBI research, and the reason behind that is to do with the biomechanics of the brain. So if you look at the brains of rats and mice, they are very smooth. They don't have like the gyri and sulci that, or the like the folds that the human brain has. But the pigs do. Pigs have folds in the brain the way humans do. So one of the hypotheses is that maybe there might be biomechanical differences if you give a TBI to this smooth brain versus if like this highly folded complex brain gets a traumatic brain injury, the way the mechanics of that tissue might be different from the smooth tissue, and that's why people are also starting to study traumatic brain injury in pig models of TBI. Um, One of the important things to consider is how this applies to a real world scenario. So, um, a lot of these models are trying to mimic what happens in people. So, if um, say someone has a fall, it's going to be probably a more focused and local kind of injury as opposed to, I'm sorry, maybe a sport concussion, which is going to be a slightly more diffused injury in nature. So, keeping all of these things in consideration, there's a number of different animal models that are devised. So, for the focal kind of injuries, there is something called controlled cortical impact, or CCI. So, that's the one that we use in our lab for my postdoc research. And then for sport concussions, there's ones where the skull is intact, and then there is a physical impact delivered to the intact skull, or a disc of a specific weight is dropped from a specific height to control for the severity of a diffuse injury. Um, there's also efforts to mimic blast injuries in the lab, so it can either be explosives in an open field, rodents, or your model organism is exposed to impact from these explosives, or um, they're exposed to kind of the pressure waves that are generated after after a blast, and that's how they try to mimic blast TBI in the lab. So there's a lot of different animal models of traumatic brain injury. You just need to select the one you use wisely based on the kind of traumatic brain injury that you're interested in studying, and the kind of questions that you're asking, or the hypothesis that you have to answer. Um, so for my research, as I mentioned, I was looking at this focused contusion sort of injury, which is the controlled cortical impact. So for this, what we did, we used rats because we had to do a lot of behavior studies as well, and rats are just more social and more similar to humans, slightly more similar to humans than mice are. So that's why we were using rats. And so for to give them the injury, we had to do surgery on them using anesthesia. So the rats are put under anesthesia, and then you have to drill a small hole in the skull, and then give an impact directly to the surface of the brain, and the speed at which the impact is delivered and the depth at which it is delivered. All of these factors can control for how severe a brain injury the animal gets. And usually you try to maintain the same injury across an animal, across each experiment, because you don't want that to become like a factor that confuses your analysis. So we were looking at some dietary supplements that we could maybe use to prevent brain damage after a TBI. So one of the compounds that we were looking at was ubiquinol, which is an enzyme that is found in the mitochondria. It's involved in the respiration cycle of the mitochondria. So the idea was that if we can improve the function of the mitochondria somehow, then all of the impaired energy and stuff that the cell goes through after the TBI might not be there, and it can lead to better health of the neurons. Another compound that we were looking at was called taurine, and This may sound a little familiar because it's something that is present in a lot of energy drinks. So if you pick up a can of Red Bull and look at the ingredients list, you're going to see taurine on it. It's actually an amino acid, but it is also used quite often as a dietary supplement because your body doesn't make it. You need to get it from outside sources. Um, so the way that we looked at the efficacy or whether how effective these compounds were in preventing brain injury was we looked at it in three ways. So we used magnetic resonance imaging or MRI to look at the damage to the structures within the brain. And the same instrument that is used to acquire an MRI can also look at changes in the neurochemistry of the brain. So, all of these metabolites and chemicals that are in the brain can be measured using this instrument, which is kind of similar to an NMR because it uses magnetic resonance or the spins of neurons to quantify the peaks that we get on this instrument using a software. So one way to look at brain damage is to see how much structural damage there is after a traumatic brain injury. Another way to look at it is to see how the chemical composition of the brain changes after a traumatic brain injury. And then the third thing is to look at changes in behavior or motor function of these animals, because you may see sometimes that there isn't as much dead tissue when you give when you give a drug. So it might prevent a drug can prevent neuronal death, and you can see this structurally or as chemical changes in the brain. But if that does not translate to improving the overall function or behavior of an organism, then it doesn't really, it's not that it's not that effective or beneficial. So those are the three things that we study in our lab: MRI, MRS, which is magnetic resonance spectroscopy, that is the thing about chemical changes in the brain, and then motor behavior of these animals, their ability to walk across a beam with minimum falls, or their ability to stay on a turning wheel, or their ability to, if we stick sticky tape on their paws, their ability to sense that there is something annoying on their paw, and how easily they can take it off. So if they're impaired, it's going to take them a longer time to take those things off of their paws, as opposed to if they are perfectly functioning. Hello, hi. Thank you all for joining in. Flowers for Algernon. I have come across so many references of that book. I have never ended up reading it. I should really put it on my list and really get myself a copy. Yeah, so that was kind of my really long spiel on traumatic brain injury. Clearly, I am very, very fascinated by the brain and by things that can go wrong in the brain. And I've been, trying to do research into how we can fix these things. If any of you have any questions for me, please go ahead and send those to me. I know we had a couple of questions come in as I was talking about the blood-brain barrier and factors that affect recovery after a traumatic brain injury. And if there's anything else that you guys would want to know, I would be happy to answer those questions, either about traumatic brain injury or research or anything. So don't be shy. This is your chance. I was curious about cases such as a guy who turned into a math genius after a brain injury. Interesting. I don't know about this. Can you do you have any more details on this? I might have to look this up. There are reports of personality changes and and a lot of these TBI patients, um, they might have a lot of so someone who wasn't really emotional before might start being more emotional and things like this. So maybe the math genius thing is just like a really it's a good thing that happened. It's a bad thing that happened to him, but ended up being good, sort of somehow. Can you speak about Phineas Gage? Oh, Jason Paget. Oh, was that the name of the patient? Okay, I will look him up. Yeah, Phineas Gage was actually a very, very interesting case in neuroscience. So I think this was um, a guy who was working on railroads back in, I don't even know what year it was, the early 1900s or something. And they had to work with, they were creating like a tunnel or something, and they were working with detonating stuff, and something went wrong and ended up detonating early, and this rod sort of went through Phineas Gage's went in, I think, through the eye and came out of his head or something like that. And he was just never the same again. So before he met with this accident, he used to be this very amicable, friendly person. And after he had this injury, he just became really unstable and aggressive, and all of these personality changes that his friends reported. And I think that was one of the early cases of people trying to understand that certain brain regions were related with um, certain characteristics and phenotypes or behaviors in people. I think something similar was the case of Henry Molaison, who was like very critical in understanding the role of the hippocampus in memory formation. Don't know how much do the medical field look into the background of the drug being used or tested upon. Background is how ecological sustainably these drugs are being produced. Ooh, that is a very good question that I do not know the answer to. I know that people are becoming more aware um, these days of the whole, sustainability and eco maintaining the ecosystem and everything being good for the planet. I don't know whether drug companies look into this. I want to say that might not be their first criteria for screening a drug. The first criteria is going to be, if the drug is effective or not in humans, because it's very hard to come by a drug candidate that is actually beneficial and has very low side effects and stuff like that. So if they find something like that, I doubt if sustainability is going to be a factor that prevents it from coming into the market. But that's a good question. Yes, I am more related to more related to neuroscience. But thank you for the question. It's always great to get this whole new perspective and think of things that you haven't really thought of. So I really appreciate that. Thanks a lot. All right. I think my time is almost up. We do have 10 minutes, but if there are no other questions, if you have questions, send them now. If not, then I just want to thank you all for being here, and thank you, Chinmaya and the Addictive Brain, for bringing me back again and giving me this opportunity to talk about something that I care so deeply and passionately about. Thank you, and I hope you all have a good rest of your day and a good weekend.